Hello, I'm Schirmer and I will be discussing the repressor Operon with you. First, I will tell you the main components of the repressor Operon. First, we will start with this red rectangle, which will represent our regulatory gene. Then we'll move on to our promoter, which is where the RNA polymerase will bind to create mRNA. And then this puzzle piece will represent our operator, which is where our repressor protein will bind. This long blue strip, will represent our structural genes, which codes for various amino acids, and in this case, because of the repressor Operon, we will be using tryptophan. We have these two puzzle piece-like shapes that will represent our repressor proteins. This one is inactive, and while this one is active. And this little blue circle will represent our tryptophan that is made and will bind to the repressor proteins. This orange will represent my RNA polymerase, and this white string will represent my mRNA that is created by my RNA polymerase. This Operon controls the production of tryptophan, and when it is usually on, but when there's enough tryptophan in the cell, it will be turned off by a repressor protein being bonded to the operator. Now I will conduct the entire repressor Operon system. First, we have our RNA polymerase bind to the promoter, and as it goes down the Operon, it creates mRNA, and then as it goes down to the structural genes, it creates various amino acids, and in this case tryptophan. When there's enough tryptophan in the cell to be able to be used, our regulatory gene will code for a repressor protein. In this case, this one is inactive and cannot bind to our operator to inhibit the production of tryptophan. So the excess tryptophan will bind allosterically to the allosteric site, and this will change the active site. So then the repressor protein will now be able to bind to the operator, which no more tryptophan will be produced. When our RNA polymerase binds to the promoter, it will no longer create any more mRNA. Our whole repressor protein, this repressor protein, is controlled by negative feedback. So then when there's enough tryptophan in the cell, it will be turned off, therefore saving energy and resources.